Hello, Maxine. Welcome to the Mind the Gap podcast. It's an absolute pleasure to have you on. In true Malone fashion, I just like to get straight into the conversation. I know you work at Genomics England, and I remember when we first met and spoke last year, I thought this is super, super cool. Could you briefly explain your role at Genomics England and your position as the project lead?
Yeah, awesome. So lovely to see you again and to be here. I'm really looking forward to this conversation. So, I work at Genomics England. I'll probably a little bit describe what Genomics England actually is because I think lots of people in the kind of health and life sciences space broadly know what Genomics England, we sometimes call it GEL, what GEL do, but it's been under kind of quite a lot of evolution recently. So GEL is a private company owned by the Department of Health, which is kind of like snazzy, new government type structure, which allows lots of different things, but one of them being agility and flexibility with which we work with different type of partners, particularly commercial partners. And it's been alive for about 10 years. It was set up to run a thing called the 100,000 Genomes Project, which aims to sequence 100,000 people with cancer and rare diseases. In the end, it was about sort of 78,000 participants, but that's because for cancer patients, they get two genomes. And once the sequencing for that was done, the analysis is very much ongoing. There's lots of people who still haven't received diagnoses for their rare condition. So that's very much still in full swing. But once the sequencing was done, there was this period of kind of soul searching and head scratching. And then what could GEL be? Because it's got all this infrastructure, it's got all these people, it's got this great asset. And so now Genomics England is the kind of platform that runs a lot of the back end to the genomic medicine service in the UK. On one hand, that's the kind of the clinical side, and on the kind of the more research side, for people who consent to their data being used for research, that then goes into kind of research asset, which is called the National Genomics Research Library, which is open to researchers, academic, commercial, UK based, international. So that's what GEL is. And then I run a program at Genomics England called Diverse Data, which is all around how we can improve equity and diversity in the genetic data we have.
That's super cool. I think just before we get into like the project that you lead, you mentioned that Genomics England is a private company, essentially, which is kind of within still a public organization. Do you think that this is beneficial for this company structure in the sense, like you mentioned, one of the benefits is agility, or is there kind of a loophole in that actually still having that public arm maybe a barrier to innovation, or maybe being private isn't necessarily the key to innovation because you still have to go public at some point?
Yeah, that's a really good question. And I think it will depend on who you speak to in the organization and what their goals are. I think for diverse data, which a lot of it is around interfacing with communities that to date have been excluded from precision medicine efforts and who are hugely distrusting of certainly the private sector, I think it can be useful because you've got a really close relationship with the NHS. On the other hand, you could also say because we are part of government, that creates a kind of different layer of distrust. So I think even for us, we kind of, in being both private and public, we serve neither side very well. I do think that so one of the, in reality, one of the ways that kind of public-private thing hybridizes and manifests is that the way that we exist in terms of the way we can spend money, how we can hire people, or the people we work with, that's all very much within the guidelines, the rules about how government works. So we still have the same procurement restrictions than if you were in the Department of Health. But when you look at the sorts of people, for example, who work at Genomics England, it's not your traditional civil servants. Yes, you have some civil servant types, but you've got, people with blue hair and funky high tops. And, it's like a very much sort of innovation-y type community and tech type community, and people who have jobs like, Scrum Master, or people who know loads about Agile, or, I'm talking out there now, but you've got like a quite different culture of people who have now gone into Genomics England, which makes it feel a bit more like a scale up than it does a government department. So yeah, that's a long way of answering that. I think that in trying to be both, it serves neither particularly well, but I think it's an interesting idea.
That's interesting because I mean, when you mentioned it, it's like the first thing that came to mind was surely that model is kind of the model to follow when we're looking at even just health tech companies in general in the UK, because you kind of sit on both sides of the fence and you can have your like almost like your tech environment, but with your proximity to the health service. But I mean, I'd never considered the other side either.
Yeah, no, and I think there's lots of different ways to make public-private partnerships and hybridized structures. And, we are, we are owned, well, us, we have a single shareholder, and that shareholder is the Secretary of State for Health. But there's loads of different types of ways in which that hybrid can emerge. So our experience is not obviously the only type of hybrid structure that exists.
Okay, cool. And the second thing that you mentioned before you went on a slight tangent was the role that diverse data plays. I mean, Genomics England, or if we're looking again using the tech analogy, like you probably have the most data in healthcare, probably in the UK. And I know that when you're formulating data sets, just even from a tech perspective, you want your data set to be representative of the entire population. When it comes to the data sets that Genomics England hold, are they representative of the UK population or any population?
Yeah. So our data is actually quite representative of the population, particularly when we're looking at ethnicity/ancestry, albeit not the same. I'm just going to use them interchangeably for this point. For gender, they're representative broadly. Socioeconomic status, not super representative. But actually, in the case of ancestry or ethnicity, which is the kind of the big data bias we see in genomics, we actually do pretty well. So that was a really important thing to, evidence and demonstrate, but also it proves that being representative is not the end point. What really matters is to what extent that affects outcomes. If you can be the most representative data set in the world, and if you've got, 0.03% of your population is, insert an ethnicity here, usually that population isn't big enough to have a sort of statistical power within a data set to really be able to have insights around them. So I never think the representativeness is a kind of good metric to anchor around on. So once we did the analysis to demonstrate that we were representative, we then wanted to do a bit of more of a deeper dive to say, okay, within that, can we find other sources of bias in our data where it might impact on outcomes that actually matter to patients or might impact, say, for example, our diagnostic performance? And after quite a lot of kind of wrangling analysis, we did find that we had some ancestral bias in our diagnostic pipelines that meant that individuals of non-European ancestry had a slightly different way in which their variants were being assessed. This is not a genomic audience for this podcast, so I'll probably stop there. But that was a really good example of, I guess, constructing your question carefully when you're thinking about looking for bias and realizing that there's lots of different ways to understand bias. It could be about fairness, it could be about equity, it could be about equality, it could be about representativeness, and all those are quite different outcomes. And it's important to attack this question from lots of different perspectives in order to make doubly sure that you are actually creating a fair, representative, and equal data set.
And what are the issues? Like if we don't representation within the data set tick, we've already ticked that box. But post representation, like you're mentioning, what are in worst case scenarios, what can happen as a result of not having that statistical power in that data set?
Yeah, so there are a couple of ways that this manifests, particularly in genomics. And if you're listening to this podcast, you know loads about genomics, you're going to be like, this is a very noddy way to describe it. But I'm going to assume lots of people don't know about niche terms in genomics. So there are a couple of kind of good canonical examples. So one is around a thing called transferability of polygenic risk scores. So polygenic scores, just think of them as any score, any risk score that you might get in medicine. And there is a kind of fairly well documented scenario whereby if a score is developed on a largely European ancestral population, and then that score is then applied to individuals of non-European ancestry, they tend to not perform very, very well. The score tends to be less accurate. And this is a general on average statement, but if a polygenic score is trained on a European population and applied to an individual of African ancestry population, the score is roughly performs 5 times as worse. Now, this kind of varies by relative and absolute sample size and trait, but that's a kind of good top line example. Another example is that one of the key reasons why we need to look at things by ancestries is that some variants in populations are rare and some are common. So in the population, you can have a kind of very rare variant, but it could result in no kind of phenotypic difference. So people might not experience any kind of ill health based on that variant. In another population, it might be really, really common, and then they might experience some sort of mutation or phenotypic manifestation of it. So what's rare or common in conditions will vary by ancestries. And if you just treat everyone the same, then you kind of you hide the fact that you've got these like very, very specific population differences. What that means clinically is that, someone, say, of African ancestry might come into the clinic and they'd be told that they've got this kind of really awful mutation. And actually, that mutation doesn't impact them at all. And they don't need to change their quality of life or their treatments or anything they do. Because actually, for people like them in that population, it's a really standard variant and doesn't make any difference to their life. So that's a kind of very, very common scenario that we see in terms of the misclassification as benign or pathogenic variants. And then the third one is that there's a thing called VUSs, variants of unknown significance. And that's that it's not even that there's a misclassification of variants, so that you don't really know if the variant does matter or doesn't matter, crudely. So that's kind of almost worse than misclassification in some respects, because you're basically giving a patient this like really ambiguous result. It's like, we just don't know. And one of the things we see in genetics is we have a much higher rate of variants of unknown significance for non-Europeans, i.e. we just don't know as much about non-Europeans. So there's this like soup of ambiguity left for many populations, often of non-European ancestry. So those are kind of three examples of like the ways in which different sorts of data biases and genetics can sort of negatively impact certain people. So yeah, polygenic score transferability, misclassification of variants as benign or pathogenic, or higher degrees of variants of unknown significance.
So, like, those are some of the ways that these things can impact certain populations. So, when we look at just genetics from a global perspective, is it currently a thing where, like, we have, say, in Europe, we do genome sequencing in one way, in Asia they're doing it in one way, in Africa they're doing it in one way, and you essentially collate all of these data sets from around the world? Or is it kind of similar to the world of academia, where there is a heavy European and like North American bias when it comes to what we understand about genomics as a whole?
I think there are loads of questions in what you just asked. So there is definitely a weird bias. So weird being Western, educated, industrialized, rich, and democratic. So that skews the sorts of research programs that run in genomics, the way that we think about populations in genomics, the sorts of methods we use, for sure. So one example of that is characterizing populations to like a really, really fine grain in terms of their ancestry. If you're going to go really, really deep in the UK, you might end up talking about like a Cornish population. Whereas we don't quite have the sample size alone in the UK to do that into the equivalent nature, say, of individuals of African ancestry. Colleagues in a major African initiative for genetics have created a ancestry ontology right down to the tribe level. And, they can do that because they've got like a really heterogeneous population. And so that's an example of a genetic kind of framework that we don't really use in the UK because that, based on the population prevalence, it might not be appropriate. But that's a kind of tool and solution that was developed within Africa in order to represent those populations. And, one of the things that we can do as, Northern European-based researchers is to make sure that we are taking those characterizations of populations developed from within those countries and making sure that we're applying them in our analysis, because so much of the analytical power is being concentrated in countries like the UK or the US. But there's also different methods that you can use to think about ancestry. So like ancestry as a concept is kind of quite wacky, because it's determined about like how far up your ancestral tree do you go. If I go one level up, then I'm Swiss and Scottish. If I go 5 levels up, I've got Ashkenazi Jew, I've got Pakistani, I've got French. And so, that what even is ancestry can be quite a philosophical thought. So there's lots of different methods you can use to kind of conceptualize ancestry in a more sophisticated way. One of those is a thing called a tree sequence, which we've been playing around with. Another one is global and local ancestry. So global being compare your whole genome to people like you, whereas local ancestry is like this little bit of your genome. This bit is compared to like people who are based in Cameroon, and this bit is for people who are based in like, I don't know, Togo. And then, you can then be much, much more specific with what you mean by ancestry for different sections of your genome. So there's loads to do from the data, from the methods to the collaboration perspective to get this right. And, we've only just scratched the surface.
So I guess that comparison of people around the world is almost like the technology that is behind 23andMe and Ancestry.com and all of these companies. Is that correct?
Yeah, so they will do a thing called genotype array, which basically is a kind of light touch way of looking at a genome. It's much cheaper, and it kind of looks at zones of your genome that we know are relevant and interesting. Genomics England does a thing called whole genome sequencing, which means we look at your whole genome, regardless of whether we know it's like relevant or not.
So, like, because I've always thought, like, I've never actually done a 23andMe test. And maybe it would have been cool if I had done it before this conversation and almost like analyzed it in this conversation. But one
Uh,
thing I've always, one thing I've heard so many times, I don't know how far true this is to say that Africa as a continent is the most genetically diverse continent in the world. And it's never made sense to me why almost the foundations of genetic knowledge in the world are not based in Africa if that is the most genetically diverse continent. Are you able to confirm or deny that fact that I found on the internet?
Yeah, I can confirm it. I can confirm it from loads of different angles. So, like a few factoids for you. There's more genetic heterogeneity within Africa than between Africa and, say, like Asian populations. Another little factoid for you is that whilst individuals of African ancestry make up 2% of genome-wide association studies, they account for 7% of the statistically significant associations, i.e., like there's much more bang for your buck in terms of like the sorts of discoveries that you can generate from individuals of African ancestry. So that's 100% true. And that goes back to the colonization of science, the Western dominance of science. And it's one of those situations that the rich get richer. The moment you get really prestigious, well-established, well-funded, new scientific initiatives and endeavors and institutions in countries or cities, then you tend to then attract the best people. Those people then attract more funding, and they just kind of get stronger and stronger and stronger as a unit. So that's why Oxford, Cambridge, London, Boston, like New York, all those places are these kind of hubs of genetic research, and they will keep becoming hubs because they've created this environment whereby the best go there, and then the best get more money, and then the best get better.
Yeah, I think it's almost interesting because in my head, thinking from first principles as someone who has no knowledge of the space, it's almost like you would expect there to be a hub, I don't know, in Kenya or South Africa or Nigeria, somewhere if we are speaking more bang for your buck. From if we're even looking at it from a business case, it seems like, well,
Yeah.
for less work, we can get more out of it. But then I think the other thing that I'm thinking about is it goes back to that thing of trust. Do you, or in your experience, have you come across almost a level of resistance when it comes to sequencing genomes of people from Africa or people from Asia, for instance?
Yeah, absolutely. And I mean, I guess there's a few ways to illustrate that. So there's there's an initiative called H2Africa, which is the Pan-African Genomic Initiative that has been running for about 10 years and was funded by Wellcome. It's now evolving into something that's a kind of a bit of a slightly new structure, but that's a kind of network of different, I guess, centers of excellence across Africa. And that's been, I think, an amazing initiative to think much more seriously about capacity building within countries, about working with African-based researchers, working with communities within Africa for the betterment of African science. But they still experience many of the same problems that like any old research does in terms of like enough resource to buy the flow cells you need to do long read sequencing, or to have the technology to do this, or to be able to store samples at an appropriate temperature that means that they don't degrade. Like all those things still remain true and still remain as huge hurdles. But what anecdotally, many people within the H2Africa network have said that, we want to create all this for us because we're tired of, helicopter science and people coming in and taking African genomes and putting into their European data banks. So actually, we're going to be really, really uncollaborative, and we're going to keep this within our African countries, or at least within the continent of Africa, because we've seen this before. That's very anecdotally what numerous people have said, and you can completely understand it that after decades of experiences of helicopter science, it's appropriate that African-based scientific institutions don't want to collaborate as much as we would love them to, having had decades of us being very extractive. From the kind of participant perspective, absolutely. So, in some of the engagement that we've done, concerns range from everything of your government. We've had this before with COVID, to what even is DNA? Are you going to like genetically change my makeup? Right to, what are you going to do with our data? How do we know you're not going to share it with the police? And all of these concerns are incredibly justified, and we have to do quite a lot to demonstrate that we are trustworthy. And actually, that can be really, really difficult because we're not starting from a blank slate. Genomics England has lots of infrastructure in place. We are structured in a certain way. We've got ways of working, and changing those ways of working, takes a long time and doesn't happen overnight. And we're all on a journey together, but we have a lot to do in terms of demonstrating our trustworthiness to these very, very valid concerns.
So at the moment, is there any measures that you are taking to rebuild that trust, or any small pieces of insight that you've found have worked versus things that completely just were not met with open arms?
I think a lot of it boils down into actually, and there's a woman in my team called Marie Nugent, who's our engagement manager, who cares deeply about research culture and changing practice to make it more accessible, inclusive, and diverse. And a lot of this is around evaluating a lot of the ways that we work and a lot of our materials. So, for example, our consent and the experience of consenting into, say, a study that Genomics England is running. And how do you make sure that that's culturally adapted? I.e., we're talking about genetic information that's resonant with lots and lots of different cultures in the way that we think about DNA. It's in a language that's accessible. It uses language that means something to the lay public, let alone different, communities or cultures or languages. And all of that is around kind of creating this body of, science is inaccessible to almost everyone for lots of different reasons. So how do we make it more accessible, and how do we make it easier for people to say, I know what this initiative is. I get why it's happening. I think it's really important and I want to get involved. But, at the same time, also, we're running an initiative on maternal health. And, one of the really big bits of feedback is that people are really concerned about joining maternal health related studies because, often people, for genetics, because some people feel that, most of their concerns with their care is due to racism in maternal care. It's not to do with genetic factors. And, one of the strong bits of feedback was that people want to have the conversation around genetics and maternal health outside of the quote unquote white gaze, because a number of our team at Genomics England are white, and actually it's quite difficult to critique the organization that's funding an initiative if everyone's sitting there, embodying many of the concerns that those populations have. So all of those are kind of just examples of things that we're working through to make sure that we're getting insights from those communities, and also trying to find ways to change the fundamental processes and structures in GEL to make them as accessible as possible.
Yeah, I think I remember speaking to a pharma company recently, and I was saying that piece of trust is like the most simple yet the most complicated part, because
Mhm.
without it, I'm like, we can't even talk about further down the line. I think one thing I've always been super interested in, generally speaking, is personalized care, and
Mhm.
almost personalized care from a perspective of, if we knew everyone's genomes or DNA makeup, we could almost tailor make medicines and different things for certain groups of the population. Now, I know we need to have the trust piece first to even get there. But in a reverse way, it almost makes me think, like, if we had personalized care, would people trust healthcare a lot more, simply because, like, your healthcare is personalized? Is there, like, what do you think of that theory? And is there room for a world where, if I don't know how it would get there, because it's almost like a chicken and the egg problem, but if we got personalized healthcare, do you think people would be more trusting? Or do you think, if there was a promise of, we are doing these studies to eventually personalize healthcare, that people would trust it more? Or
Mhm.
would they mistrust it even more, almost like, well, if it's personalized, now you guys are going to poison us and do something good for them? I don't know if that makes
Oh,
sense.
man, that's such a good question. So I think personalized healthcare in the way that you're talking about it includes a few things. One is the underlying, let's say, kind of more clinical phenotypic data. And that means like everything that's like in your medical records, right, to administrative data, like your social determinants of health data, where you live, where you work, that kind of stuff. I think there's a component of obviously your genetic data, which allows some degree of personalization. There is a slightly tropey quote, which is that your postcode is more informative than your genetic code. So if you're thinking about personalized medicine, let's just be cognizant of where currently a lot of the personalization happens. And it's probably not at this point necessarily in your genome on average. But I think a third component of that kind of data piece is the human component in terms of the care you receive and who you're receiving it from. There was I saw a kind of play a year ago called The Doctor, and there's a kind of line in it where it says, it's not viable to have clinical staff completely representative of patients, because you'll then need fat doctors for fat patients, Jewish doctors for Jewish patients, black doctors for black patients. And I kind of agree and disagree in equal measures, but, personalization also partly comes from the environment in which you're receiving care and who you're receiving it from. So I think you need all of those three things in place in order to get actual experiential personalized care, not just like scientifically determined personalized care. But I do think you put like an interesting provocation, which is, if you claim to be personalized and it doesn't work for someone like you, when it doesn't work for you, then you're like, well, it's not that personalized then. Whereas I do think that there is a kind of acceptance that the NHS is serving the population on average. And therefore, if you are in the tails, you're on the outskirts of that average, or you're on the edges of that average, which completely sucks for people who are on the average. So we're not sorry, people completely sucks for people who are on the tails. But we kind of understand that we're in a national health service, and therefore you're going to get tails in all these different angles. So yeah, I think my main point is that at the moment, personalization doesn't come from genetics. It comes largely from all the other data that we are not using appropriately, particularly clinical data that has so much richness in it that's currently untapped.
On the note of data, sorry to just bring it back. I remember reading something about how if you are I'm from Zimbabwe. And if you are from Zimbabwe, if you do a 23andMe or like any of these genome studies, your results are not accurate, and it will always show up as Kenya as being like your place of origin. And that given due to like Zimbabwean people, a lot of Zimbabwean people derive from like the Bantu people, and the Bantu tribes can be found from Kenya all the way down to like South Africa. Is there things like this across the world where, like, when we are trying to go, like what you're talking about ancestry, it might sound extremely dumb, but it's literally how my mind is just processing the information. Is there a world where this information almost can be wrong? So, like if I do a 23andMe study and it comes up as Kenya, but I actually don't have any link to Kenya, but I just come from the Bantu people, and almost the data that we have on that group of people is just very like spaced out across a massive area, that actually this is not specific to me, but just a generalistic approach, if that makes sense.
Yeah, no, that totally makes sense. And I think that's because ancestry is quite a complicated concept that because we don't see ancestry in our day-to-day lives, it's not something that we are like regularly engaging with as a concept. We, as Genomics England and 23andMe and lots of other institutions, try and create proxies to make it more understandable. So your country of origin is like a kind of proxy for ancestry. But obviously, ancestry, as you correctly said, in this context, is picking up Bantu populations, which are disparate in certain parts of Africa. So I think that the country thing, it just helps people anchor at what ancestry means in terms that make sense to them today in the way that the world is structured. Just like ethnicity is not the same as ancestry, but can sometimes help people understand that the two are somewhat related in terms of, like, if you are of African ancestry, you are obviously more likely to be Black, but that doesn't mean that you're Black, and those things are not the same. So all of these are just proxies to help us understand what is quite a complicated concept of ancestry. This also is partly determined by how granular you want your ancestry to be. So for example, when we started the program at Genomics England, people were grouped into 5 continental ancestral groups, Europe, Africa, etc. And we've been working to make that more and more granular. And we get these like very specific pockets of concentration of populations in East Africa, West Africa, Southern Africa, and North Africa. And if you were to make it even more granular and go from 5 categories to 20 categories to 50 categories, you obviously start to get more specific. I don't know how specific 23andMe is, but I think that it's one of those things where the more people they have from those regions, the more granular they can go.
Yeah, I guess so. And I mean, I'm assuming their dominance is more in like the Western world as opposed to in Africa or in rural parts of South America, for instance.
Sure, but also interesting at the same time, 23andMe is the largest non-European data set in the world as well. So even though the majority of 23andMe's users or customers or whatever way they're called, people who've done it are going to be white European, in total terms, it's still the largest non-European data set in the world of genetics. And that's there's something in that to be impressed by.
So that's 23andMe is like an example of another institution or another company that has kind of taken this approach of almost analyzing the world's data. How can other institutions, whether that be like universities or companies or public-private hybrid organizations, ensure like diverse participation in genomic studies generally?
Oh man, it starts at every single level. So I think that, I think quite easily you can get overwhelmed with the fact that it's all sort of, structurally racist and we're never going to solve this by lunchtime. And I think there's a kind of realism of, okay, what can we do within our power, within a reasonable time frame that means that we can take one step towards making this slightly more diverse and more equitable. So the way that we think about it in the Diverse Data Program at Genomics England is that you've got pipelines of research that starts with like how the funding is structured, how the questions for research are put together, how the studies are designed, how the models are developed, how those are applied, and how those are evaluated. That's like a kind of standard research pipeline. And each step of that research pipeline might introduce some bias. So, for example, we are creating a massive whole genome sequencing data set for sickle cell. But if we create this data set and then we unleash it to the world of researchers, and researchers just answer the same questions they've already asked and answered, you might not necessarily start answering questions that matter to patients in a way that's kind of potentially cultural paradigm shifting. So one of the things we're doing is we're doing a collaboration with the James and Linda Alliance and the Sickle Cell Society to create a patient-centered top 10 priority list of research questions. So that means that in 18 months, we've got like a lovely fancy sickle cell data set, but we've also got a whole research agenda that's been written by patients with sickle cell to say, to the research community, these are the questions that you should be asking because this is what matters to us. Don't be doing your like random little methodological stuff. Do the thing that matters to us in our care. So it's not just a data problem. It cascades through the whole research process. So that was just like one example of something that's not data driven for us, but we're just trying to make sure that we're thinking more holistically about the environment in which data is made as well.
Could you give an example of like what sickle cell patients, for instance, have asked, if it's shareable?
Yeah, no. So kind of early insights already is that, so obviously the genetic driver of sickle cell is very well known. That's not something that needs to be discovered or investigated. We know why people have sickle cell, but what we don't know is why there's the genetic drivers of kind of symptom heterogeneity, i.e. why some people experience one symptom and other people don't, or why some people have like a very acute version of a symptom and other people don't. So a really good example is pain crises, why even within the same family do some people have huge pain crises and other people don't. So that's one of the examples of the questions that really matters to people, which is, I want to not be experiencing pain. I don't understand why my brother has pain crises and I don't, or the other way around, to be honest. And what can we do to understand why there's such substantial differences even within a family is like the sorts of questions that people are saying are of importance to them. I guess there's a really interesting kind of challenge in all of this, though, is that we are talking about genetics. It's not about direct care. And so obviously, if you take some person randomly on the street who's got sickle and you said, what can genetics do for you? A lot of the time it is very care focused, and a lot of the genetics research stuff is quite upstream. So that's going to be like a tension for us to balance, which is how do we make sure that we, hold true to ensuring that it's a patient-centered research agenda, whilst making sure that we bring people who engage in this process up to speed enough on genetics that they can both like understand what they need to understand and then inform us with what sort of research we should be doing. I think that's going to be quite like a difficult balance to strike.
No, yeah, I think I remember like when I first met you and had a conversation with you, I think probably the thing that I admired the most is I think you're one of the people that I've spoke to in the space who was looking at the problem from a holistic perspective. A lot of the time people just want to attack like the small thing without realizing that actually we need to go a bit further upstream, look at the trust, look at the funding, look at how we are devising the questions. So I personally get very excited about the work that you do because I'm like, well, if you've thought all of that through, it's just a matter of allowing like the dominoes to slowly fall, and eventually like things will hopefully get to a place where they should have been. Just before we close, I just wanted to find out from a place of curiosity, like whilst you've been leading the program at Genomics England, what has been like the most significant learning point or like revelation in advocating for diversity in the field whilst you've been in your role?
Oh man, it has been 2 years of insane learning curves because, we received the funding and then, there wasn't a team. We had to grow the team. We had to devise a strategy and we had to deliver all at the same time. So it's been a sort of wild 2 years. I would say that the main challenge / revelation / I think thing that makes this particularly complicated is that initiatives like Diverse Data, of which there are a number across the world in different countries, are crudely, broadly divided into initiatives that are kind of scientifically driven and that they think they are about characterizing human genetic variation. And then ones that are more equity driven, which is around kind of engagement first, looking at trust and looking at, I guess, structures of power and influence within genomics and how to make sure that everyone has as equitable experience as possible. It's a false dichotomy, but just to illustrate the point, those are kind of how they're divided. I think that within Diverse Data and within the ecosystem and within the organization and within the Department of Health, there's loads of different opinions about whether we are one or the other. And there's not a wrong or right answer, but they do incur fundamentally different strategies and different approaches. And so I think that one of my revelations is that like linking the kind of social science, trust, engagement stuff with the purely scientifically driven strategies is very, very challenging. And those are fundamentally different languages. And actually forcing them together, or more importantly, getting the engagement side to inform the scientific side is a real challenge if you're trying to also do that whilst building a team and delivering at the same time. So I think that's probably the one that I've kind of found most interesting and most challenging in equal measures.
Yeah, I think it's that hard line or that hard middle ground between the social science and almost like stereotypically like hard science is always even for me, like it's always difficult to explain like why people will behave in a certain way without using both sides of the fence. But then both sides of the table will argue that actually this is hard science. Like, how can you use anecdotal evidence to say that like this is what's happening? And then the other side of the table says exactly the same thing to say, actually, well, your hard science doesn't even include us in the studies. So how do you know that our anecdotes are not correct? And yeah, it's just finding that middle ground and
no, completely. And I've been caught in the crossfire of that. And, you know, I try my best to smooth over those like different paradigms of working. But, you know, there are people within the team and at Genomics England who are so passionate about both sides. Actually, it's just about like how you harness that passion to be synergistic rather than, you know, working against one another.
Yeah, 100%. And as a closing tradition on this podcast, I think this conversation has been super interesting. Literally, I know I'm going to go back and listen to it, and I don't usually listen back to episodes.
There's no way I'm listening to my own voice for half an hour.
I'm exactly the same. Like, even when I'm editing this podcast, like I almost have to cringe because I have to listen to myself speak for like 40 minutes just to be able to edit it. But as a closing tradition, one thing I always ask people before we go is, if you could improve healthcare for your community, how would you improve it? And your community can be absolutely anything you want. You just want to know who your community is and what would you specifically do?
I find this was a very, very hard one. And you told me you were going to ask it 45 minutes ago, and I still don't feel prepared. So I'm going to half answer it with an anecdote from a friend of mine who ran a beauty business. And she basically had the wonderful idea, and actually we've seen equivalents of this in different sectors, but she had the wonderful idea, which is there's a huge issue with women not turning up to their smear exams, and particularly not white women and women from deprived backgrounds. And for her, the nail bar is like the center of female community in many parts of London, certainly for her. And so she was like, why don't we train nail technicians to also be able to deliver smear tests? You know, get women whilst they're doing their manicures to also do their smear tests. And I guess stuff like that of like in community opportunities for interventions or screening or conversations don't usually happen. I like my favorite types of anecdotes. I think we hear examples of that as well. It's like male barbers talking about mental health as well. It's like another example we've seen that's happened a bit. So yeah, I'd get more women to turn up to their smear exams by getting them to be co-located with their manicures. Is probably what I do.
Okay, cool, amazing. That's probably the most unexpected answer that we've had so far. But okay. I love it. Like, yeah, like there is a company in America which is like a male barbershop, and they basically deliver health information like prostate cancer, mental health. And I always think things like that are super cool because it's like the middle ground between the community and something that people don't trust. And if we are talking about trust, I guess people trust their nail technician.
Yeah. Although she says having had I bit my nails, you can't see them on podcast, but you can see them on video. But I bit my nails so badly the other day I had to have an emergency manicure because it felt like a health intervention because my nails were so raw. Anyway, too much information on that note.
Fair enough. On that note is a great place to finish. But thank you so much, Maxine. If anybody wants to find out more about the work that you're doing at Genomics England, how can they find out? Where should they go? And how should they stay in touch?
Yeah, so if you go on to the Genomics England website and you just look at diverse data, then there's information about the program. There's also our kind of generic email address. I think my email address is literally all over the website anyway. And also, if you are interested in talking more about the more academic, researchy side of just health, data bias, AI bias in health, then I think my Alan Turing Institute profile is public as well with my email address there. So if you want to talk to me and nerd out on the data front, I love to get those sorts of emails.
Amazing. Well, thank you so much for your time, Maxine. It's been an absolute pleasure.
Thank you so much and lovely to see you. Bye-bye.
Bye.